In recent years, the world has faced firsthand the devastating impact that viruses like COVID-19 and Ebola can have on our communities, economies, and daily lives. The rapid spread of these viruses has shown us just how crucial it is to find effective treatments against both known and emerging threats. That's why efforts to combat these viruses are more important than ever for humanity. Hello, everyone, and welcome to our podcast. I'm Omar, and today we have the pleasure of speaking with Dr. Marceline Côté, an associate professor and the director of the Center for Infection, Immunity, and Inflammation at the University of Ottawa. Her research is focused on understanding how these viruses interact with human cells, and ultimately on developing treatments that can stop their spread. Today, we're excited to dive into her fascinating work on sphingosine kinases and using them to combat viruses like Ebola. Welcome to our podcast, Dr. Côté. Thank you for coming.
Thank you very much for the invitation.
To give everyone a quick overview, Dr. Côté co-authored a paper that explores an innovative approach to stopping viruses which is akin to outsmarting a Trojan horse. Imagine if the defenders in Troy knew that there were soldiers hidden inside the wooden horse and allowed it to enter the city anyway in order to set up their own ambush. In a similar fashion, Dr. Côté and her team have been studying how using inhibitors of sphingosine kinase, also known as SK for short, could block viruses like Ebola from causing any harm to our cells. With SK inhibitors, the virus still enters the cell but is rejected before it can do any damage. This approach was tested on a range of viruses, and it might be the beginning of a whole new type of broad-spectrum antiviral treatment. So, our first question to you, Dr. Côté. Tell us about how viruses like Ebola enter our cells in the first place.
Thank you very much. Very good question. I really like your analogy with the Trojan horse because that's a little bit like this. So, Ebola virus and other viruses are what we call envelope viruses. So, they have a lipid envelope, and at the surface of this lipid envelope, or inserted in it, you've got glycoproteins, so viral fusion proteins. And these are really important to recognize the cell and then do a bunch of things to allow it to enter and deliver its genetic material into the cell. And in addition to this, it's also disguising itself. So for Ebola virus in particular, in the lipid envelope, you also have a type of lipid that we call phosphatidylserine. And that's actually a lipid that is only exposed on cells that are undergoing cell death, apoptosis. So we call this apoptotic mimicry. So it's making it look like it's an apoptotic cell or apoptotic bodies, and basically macrophages, which are primary targets of the virus. So these are immune cells that are going around our body to clean up apoptotic bodies, for example, but also microbes. They detect those and they just want to eat them, right? So the virus is tricking those cells that way. So the virus attached to the surface of the cell, it will trigger multiple different signaling pathways to allow the virus to be internalized. So it's, a little bit like eating. Mm-hmm. And then it has to traffic through what we call the endosomal system. So it's a little bit like when a cell takes nutrients, it has to take it up. It's going to go into what we call endosomes. And there you've got the receptor for Ebola virus. So it goes inside, it traffics, and then it finds its receptor attached to it. And then this will trigger what we call the viral fusion protein to allow the merging of the viral envelope with the cellular envelope, and then the genetic material goes in it. So that's what happens. So it's very complex, actually. So you've got multiple different steps, and all throughout this, journey, you've got a lot of cellular proteins that are essential for this process. So in my lab, that's why we're interested in understanding how all of this happens, because if you find some interesting host cellular proteins that are required, then you can potentially target them to inhibit viral entry and stop the Trojan horse, indeed.
Yeah. Okay, that's really interesting. So I want to kind of ask you about sphingosine kinases, which your paper talks about. So what are they and why are they a good target for blocking viruses?
Yeah. So again, a very good question. So in our attempt to try to find these cellular proteins that are involved in viral entry. We did a screen of small molecule inhibitors, which had known targets. And that's how we found the sphingosine kinases as being potentially involved in viral entry. We had, of course, to validate all of this. But those are proteins, so enzymes that will phosphorylate sphingosine. So they will add a phosphate to sphingosine, and this is a signaling lipid. So as I said, during viral entry, the virus will induce a signaling cascade that's important for the internalization, so they're eating, but also the trafficking of the virus to the receptor. So to get to the receptor. And we think that sphingosine kinases are involved in this through the phosphorylation of this important lipid.
Okay, wow, that's really interesting. You also mentioned that SK inhibitors were tested on different types of viruses in the paper. Could you walk us through how effective this approach was across the different types of viruses and what kind of results you were getting for the different kinds and if there were any surprises or interesting findings?
Yeah. So one of the things that we had to do first was to understand the mechanism of action of these inhibitors. We had to make sure that the targets were indeed sphingosine kinases, and we did that. And then figure out where in the pathway. As I said, it's very complex. So you've got a lot of things happening: the eating, the trafficking. So we have multiple assays to look at this, and we found that it was really the trafficking that was blocked by these inhibitors. So the sphingosine kinases are involved in the trafficking, which is also a relatively new thing. In the literature, it's mostly for internalization, things like this. But what we found, it was for trafficking. So we're going to follow up on this. So we still don't understand it fully, but it seemed to be trafficking. So then the question is, for other viruses that also requires this trafficking to endosomes and late endosomes, could these inhibitors work? Right? So that's exactly what we did. So we have a platform in the lab to test entry of various highly pathogenic viruses. So of course, Ebola virus, we don't work with Ebola virus in Ottawa. So it's not the real virus, replicative virus. We have a colleague at Winnipeg who does this. But we use what we call pseudotypes. So it's a little bit, again, the Trojan horse kind of idea. So we take a safe virus and then we decorate this virus with the glycoprotein of the pathogenic virus. So we do this for Ebola, but we can do this for multiple other viruses. And that's what we did. So we tried other hemorrhagic fever viruses, such as Lassa fever virus, as well as Unin virus. You may not know necessarily those viruses, but they also have receptors that are in endosomes. We also tried Nipah virus, influenza virus, and we saw that for those that needed to go deep in the endocytic pathway, so in the late endosomes and lysosomes, the inhibitors were also working. So Lassa fever virus was inhibited. We did also SARS-CoV-1, which was inhibited, and our colleague also tested SARS-CoV-2 in some cell lines. It was also inhibited. So it was really the whether or not the inhibitors would work really dependent on the entry pathway of the virus.
And based on those results, those findings that you had, what would you say is the future of broad spectrum antiviral treatments? Could SK inhibitors potentially pave the way for more of these types of therapies, or are we kind of looking for something else?
Yeah, no, I think for broad spectrum antivirals, more and more people are looking for host-directed approach. So a lot of the inhibitors that we have, which are really good, of course, are virus-targeted molecules. So for example, an inhibitor of the protease of the virus, if it has a protease of the polymerase, of so very, very targeted, that doesn't allow you to do for these inhibitors to work against a lot of different viruses, of course. But if you're targeting something from the host that is required by multiple viruses, such as sphingosine kinases in this case, then you've got an opportunity to be able to have an antiviral that is broad spectrum against many viruses, such as the one we tested, but also viruses that we don't that doesn't have emerged yet, right? So to be prepared. Of course, we need to test this, but I think there's more and more labs that are looking into these OS-directed approaches for pandemic preparedness, mostly.
Yeah, that sounds like a very promising avenue for investigation. But one thing our listeners might be a little curious about is the safety of these kinds of therapies. So, are there any known side effects of using SK inhibitors, or maybe any concerns that we might have about how they might affect cells outside from blocking the viruses? What has your research shown on that front, really?
Yeah, so this is very important. Of course, safety is essential for any of these um, potential antiviral. So just so you know, so these viruses that we're targeting are viruses that we call causes acute infection. So it's something that is really fast. So if you would be given the antivirals, it would be for a relatively short time, but it remains that it needs to be safe, of course. So the inhibitors that we have tested are FDA approved already. So the safety concerns are not an issue, obviously. They have been tested in clinical trials already, but this is something that needs to be done every single time. And then in terms of what these inhibitors can do, so some of these are being used for cancer. There's some it's also for the immune system as well. So this is something to take a look at, obviously. So more work needs to be done. Obviously, we one of the next step would be the animal studies first. But yeah, no, this is something we keep in mind all the time.
Yeah. As with any development of new therapies, I'm sure the safety process is very long and rigorous.
Yes, yeah,
But in
but it
theory,
needs to be done.
yeah,
Hmm.
if it goes well and this therapy proves to be effective in the long run, how do you think it could impact public health responses during viral outbreaks, especially for viruses like Ebola, where quick responses are critical?
Yeah. So again, I think it's about preparedness, right? So when you have a new virus emerging, if you have already a tool set or an arsenal of these potential OS-directed antivirals that are safe, so that this has to be done already, that are safe, that we would know what would be the dosage and everything, we could quickly respond to any new emergence of a virus. And especially what is really great about the small molecules, these are usually stable. Many of those can be given orally as well. So it's easier to deploy.
Wow, it sounds like SK inhibitors could be a real game changer in this kind of field. Yeah, that's all the questions we had for you today. Thank you so much for joining us, Dr. Côté, and sharing your thoughts and insights. The idea of stopping viruses like Ebola before they can do harm is very inspiring. And I think we'll all be watching very closely as this field continues to develop. And for our listeners, thank you for tuning in. And remember to stay curious. Thank you very much, Dr. Katar.
Thank you.